let's start our new chapter, that is biomolecules. So in this chapter, biomolecules means what? Biological molecules, correct? So because we are going to talk about all the essential molecules that are present in our biology, that are required for any living organism to live, so we are going to discuss about those. Basically we are going to talk about chemistry. This chapter is going to involve lots of chemistry. So there are wide diverse types of living organisms, right? Whether it is in our biosphere, if you see, in our biosphere means in our earth basically. So when you look at the earth, you can see there are lot of varieties of living organisms. So the question that comes in our mind is, are all the living organisms made up of same chemicals? Whatever living organisms we have, are they made up of the same chemicals, elements and compounds? Are they made up of same type of elements or and compounds? So in chemistry we learned about how elemental analysis is performed, how we do the analysis of different elements. If we perform such analysis on plant tissue, animal tissue, or any microbial paste, we can obtain a list of elements like carbon, hydrogen, oxygen, and many others. So, irrespective of content per unit mass of living tissue. So, we can find out their respective content. How much content is present? How much is the content that is present? The content of hydrogen, carbon, and oxygen and other compounds, in how much content they are present per unit mass of living tissue, we can obtain those different compounds. So if the same piece of analysis is performed on Earth's crust, if we do the same analysis on the crust of the Earth, as an example of non-living matter, then we obtain a similar list. We get a similar list because even the Earth contains different kinds of compounds, right? So now, what are the differences between the two lists? In absolute terms, no such differences could be made out. In Earth, whatever compounds are present, same type of compounds are present in our living body. So, all the elements present in the sample of Earth's crust are also present in the sample of living tissue. So, our living tissue is made up of nothing but it's a part of Earth's crust only, right? In the Earth's crust, whatever we have, same kind of things are present in the living tissue. This is a very important line. So, now a closer examination shows that relative abundance of carbon and hydrogen with respect to other elements is higher in any living organism than the earth crust. So when you talk about living organism, they have more carbon, hydrogen with respect to other elements. If you compare with respect to other elements, hydrogen and carbon is more in any living organism. That's why these are known as organic, right? Organic compounds are what? When you talk about what is organic and inorganic compounds, two type of compounds we learnt: organic and inorganic compounds. Organic compounds are nothing but they contain carbon and hydrogen, so they are nothing but mainly hydrocarbons. They must have hydrogen and carbon in that, whereas inorganic compounds are other elements that don't contain this hydrogen and carbon. So now let's talk about, so generally when you talk about living matter, organic compounds are present in living matter. So now how do you analyze? Okay, all the story is done. Good, very nice story. What is present in our living body is almost similar to the earth. But how did we find out that? So there is a method for finding out that. To get the answer, one has to perform chemical analysis. We need to do chemical analysis. So how is this chemical analysis done? Let me tell you. So, to perform this chemical analysis, first we take our living tissue. Whatever living tissue is there, whatever compound you need to analyze, we take that. Then we mix it with trichloroacetic acid. After mixing it with trichloroacetic acid, we grind them. Okay, we grind it with trichloroacetic acid. So both of these together are grinded. They are ground together or grinded together. We grind them together. Okay, we mix them using mortar and pestle. Then what we do? A paste is formed. So when you mix both of them, you get a paste. that paste is filtered. We filter the paste. So when you filter the paste, you have one liquid pool. Correct? When you filter it, one side you will get liquid, and on the filter paper or on the cotton, whatever you use to filter it, on the cotton, another kind of semi solid which is not passing through that, which this does not pass, right? This does not pass through filter paper. So this is known as our, this liquid is known as our acid soluble pool. Acid soluble, because we mixed it with trichloroacetic acid, and trichloroacetic acid is acid, right? So whatever was soluble in this acid comes out through filter paper in the form of liquid. That is known as acid soluble, whereas the one that does not come is known as acid insoluble pool. So generally all the macromolecules, whatever big molecules you have, they are a part of this. They are known as macromolecules. These are known as macromolecules because they are bigger molecules. Whereas if you talk about these are known as micromolecules. So this way we can separate them, right? We get two compounds. One is macromolecules and other is micromolecules. We can identify macromolecules are bigger molecules and micromolecules are smaller molecules. And how do you separate organic and inorganic compounds? These are the ways to separate big and small compounds. See, macromolecules are generally big molecules like proteins and other whatever big molecules you have, whereas when you talk about macromolecules are nothing but smaller molecules like amino acids. They are small. Like if you talk about simple sugars, all those are smaller compounds. So now let's talk about that in detail. So whatever I told, we can take any living tissue. It can be a piece of liver, vegetable, whatever, and grind it in trichloroacetic acid, which is Cl3, trichloro, right? So Cl3 CCOOH, because acetic acid contains COOH acid. So using mortar and pestle we grind them. Then you will get a thick slurry. Now if we strain this thick slurry through a cheesecloth or cotton, you will obtain two fractions. One is called the filtrate, and or more technically the acid soluble pool. So filtrate means what? This is known as our filtrate, the thing that comes out of filter. After filtering, whatever you get outside, that is known as our filtrate. And whatever is not getting filtered, so basically whatever does not get filtered, that is acid insoluble pool, that is called retentate because it is staying there, or acid insoluble fraction. So scientists have found thousands of organic compounds in the acid soluble pool. In the acid soluble pool we get thousands of organic compounds. So we are going to analyze a living tissue simple and identify particular organic compound. It will be sufficient to say that extracts of the compounds then are subject to extract of various separation techniques. One has separated the compound for all other compounds. In other words, one isolates and purifies a compound. We can separate the compounds by doing different kinds of techniques. Analytical techniques, when applied to the compound, give us an idea of the molecular formula and the probable structure of the compound. Using analytical techniques we can find that. So all the carbon compounds that we get from living tissues can be called as biomolecules. So that is the meaning of biomolecules. That is the chapter that is known as our biomolecules, all the organic compounds that we are going to learn. But living organism can also get inorganic compounds, right? It has inorganic elements also and the compounds in them. So how do we know this? A slightly different but destructive experiment can be done. So in this experiment, what you do? First experiment, we identified between macro and micromolecules. Macromolecules and micromolecules we separated. Whatever are the bigger molecules and smaller molecules we separated using trichloroacetic acid. Now in this experiment what we are going to do, we will take a tissue and dry it. whatever we want tissue, we dry it. Okay, any organic compound, we'll dry it so that all the water content goes away. First we remove all the water from it. So all the water we need to remove. Next, what we do, we'll burn it. If you burn it, the ash that is left over, that ash is inorganic. It contains all the other inorganic compounds, because when you burn it, whatever carbon and hydrogen was present, they become carbon dioxide and water vapor and escape, right? They will become carbon dioxide and water and escape. So whatever is the ash or other inorganic compounds that are left over, they will be present. So that's why it is called destructive experiment. So you can weigh a small amount of living tissue and you can dry it. So all the water evaporates. The remaining material gives the dry weight. Now you will come to know what is the dry weight. Now if the tissue is fully burnt, all the carbon compounds are oxidized to give gaseous form. That is carbon dioxide and water, whatever I told. Hydrogen forms water and carbon forms carbon dioxide and are removed. What is remaining is called the ash. The ash contains inorganic elements like calcium, magnesium, etc. Now the inorganic compounds like sulfate, phosphate, etc., these are also seen in the acid soluble fraction. When initially we prepared the acid soluble fraction, right, that is our micromolecules there. Therefore, elemental analysis gives elemental composition of living tissues in the form of hydrogen, oxygen, chlorine, carbon, etc. While analysis of compounds gives an idea of the kind of organic compound and inorganic constituents present in living tissues. So you can see here, this is the comparison. So when you talk about in the percentage of Earth's crust, how much of what compound is present. So if you see, second highest compound in our body is carbon, and oxygen is the highest in our body. We have highest is oxygen. Why we have oxygen highest? Because we have H2O, water. Lot of percentage of our body contains water, and it has oxygen also, because our cells require oxygen, right? So hydrogen is only 0.14% weight of the Earth's crust, because hydrogen is very light. That does not mean hydrogen is very less, okay? Because hydrogen is very light. In our human body, it is 0.5%. Carbon is 0.03% in the Earth crust, whereas in human body it is 18.5%. Oxygen is 46%. This is 65% in human body. Very little nitrogen is present in human body. It is 3.3, 0.03, whereas 0.3 is sulfur. Then when you talk about sodium is 2.8%. In human body it is very little, 0.2%. Calcium is 3.6, in human body 1.6. Magnesium is 2.1, in human body it is 0.1. Silicon in the earth, large amount of silicon, because the soil, you know, silicon is mainly the soil and all contains lot of silicon. It is made up of silicon only, which is negligible in human body. Now when you talk about sodium, formula is Na plus, K plus is potassium, calcium is Ca2 plus, magnesium is Mg2 plus, H2O is water. Compounds, other compounds are NaCl, CaCO3, PO3, phosphate ion, sulfate ion, all those different types of ions are present. Now living tissues. From a chemistry point of view, one can identify functional groups like ketones, aldehydes, aromatic compounds, etc. All these are present. From biological view, we will classify them into amino acids, nucleotide bases, fatty acids. So based on these, we are going to study in biology. So amino acids are organic compounds containing amino group and an acidic group as substituents on the same carbon. So alpha carbon, hence they are called alpha amino acid. They are substituted methanes. There are 4 substituted groups occupying the 4 valency positions. These are hydrogen, carboxylic group, amino group, and a variable group designated as R group. Based on the nature of R group, there are many type of amino acids. So when you talk about amino acid, what is given here, I will just tell you. They are called as alpha amino acids. The alpha carbon, hence they are called alpha amino acid. Why alpha amino acid? They are substituted methanes, so they have alpha carbon. Okay, so we have one carbon, and on that carbon we have COOH, that is our carboxylic group, and along with that we have our NH2. Okay, so this is how the structure of our amino acid looks, and we have one variable group, that is R group, and along with that we have our hydrogen. So this is what is containing. So there is hydrogen, as I drew here. We have hydrogen, we have a carboxylic group, then we have a amino group, then we have a variable group, and this is how the basic structure of amino acid looks like. So all the amino acids that are present in our body are going to be of this kind of structure. So this R group will be varying, and in some places hydrogen can be replaced by something else. So however, those which occur in proteins are only of 20 types. So in our body we have amino acids, but they are mainly in proteins. When you talk about, they are only 20 types of amino acids. The R groups in these proteinaceous amino acids could be hydrogen. The amino acid is called glycine. So when we have hydrogen here, so when the R group here, basically if we have hydrogen, see, this is how the structure of that will look like. Carbon, instead of R group we have hydrogen, so we will have two hydrogens here, and this becomes COOH, and this is our NH2. So this is called as glycine. Such kind of amino acid is known as glycine. A methyl group, alanine. So instead of this, if you have methyl group now, if I write here CH3, then this becomes alanine. And if I replace that with hydroxymethyl, if I replace it with hydroxymethyl, then it becomes serine. So the 3 of the 20 are shown in figure 9.1. In figure 9.1 you can see 3 of the 20 amino acids. So CH2OH is our serine, that is hydroxymethyl. If CH3 is present, it's alanine. If only hydrogen is present, it's glycine. Now the chemical and physical properties of amino acids are essentially of the amino, carboxyl and R functional groups. Based on the number of amino acid and carboxylic acid, they are acidic. They can be acidic based on how much amino acids they have. So this carboxylic acid and this is amine, right? So if this NH2 is more, if some compound is having more NH2 groups, that becomes more basic, and if COOH is more, it is more acidic. So depends on what is more. So for example, acidic if you talk glutamic acid, basic if you talk lysine, and neutral, this is our neutral because it has one NH2 and one COOH group. So this is such example of that is one is valine amino acid. Similarly, there are aromatic amino acids like tyrosine, phenylalanine, tryptophan. If they have aromatic group, that is benzene ring. A particular property of amino acids is ionizable nature of NH2 and COOH groups. Ionizable nature means they can form ions. Hence, in solutions of different pH, the structure of amino acid changes. So you can see here what happens. So basically, this is the structure. This H group, basically from here, our H has come here. Okay, and this is known as a Zwitter ion. This kind of compound is known as my Zwitter ionic form. In this form, the amino acid will exist. Now, based on whether it is present in acidic pH or basic pH, for example, if it is present in acidic pH, in acid what is more? H plus ions are more. So if H plus ions are more in acidic pH, so what will happen? Here it forms this kind of structure because here an H extra H gets attached. Whereas in water what happens? H will leave from here and it forms in basic pH it along with OH minus. In basic pH OH minus is more, so there one hydrogen plus goes and attaches to this and it forms H2O, right? So basically this is the structure which is found in basic pH. I'll just tell you. This is my Zwitter ion. In basic pH, this is what happens, and in acidic pH, this is what is the situation. So you can see there. Hence, in solutions of different pH, the structure of amino acids changes. Now, lipids are generally water soluble. We spoke about amino acids, now let's talk about lipids. Lipids are generally water soluble. They could be simple fatty acids. A fatty acid has carboxylic group that is attached to R group. The R group could be methyl, ethyl, or larger number of CH2 groups. 1 carbon and 19 carbons. So from 1 carbon to 19 carbons can be present. For example, if you talk about palmitic acid, it has 16 carbons including carboxyl group, and arachidonic acid has 20 carbon atoms including the carboxyl atom. So fatty acids could be saturated without double bond, or they can be unsaturated with more two or one or more double bond. So if they have double bond, they are called unsaturated, because saturated means what? Saturated means they are full, right? For example, this structure methane, ethane, if you see, ethane will be called as saturated because there is no double bond here. But here, if you see, here there is double bond. Double bond means what? Another compound can come and attach here, correct? So that says us that shows us that it is not saturated. Saturated means full. Okay, it is not full because there is an empty space, double bond. Another simple lipid is glycerol, which is trihydryl propane. Many lipids have both glycerol and fatty acids. Many lipids have both glycerol and fatty acid. I will show you the structure of fatty acid. So this is the structure. CH3 and COOH will be present. That is why it is called acid. And in between it can have how much ever carbon chain, large carbon chains. And this is one example of glycerol. It has OH, OH, OH. This is how the structure will be. C carbon chain will be present, and along with that OH groups will be attached. Here the fatty acids are found to be esterified with glycerol. With glycerol it is attached. They can be then monoglycerides, diglycerides, and triglycerides. These are also called fats and oil bases on melting based on melting point. So fats means they are solid generally, like ghee and all, and oil they are already in liquid state, right? So they are called as oil based on melting point. What is the lower melting point? So oil have lower melting point, that's why generally they are present in the form of liquid in natural temperature, and hence they remain as oil in winters. Can you identify fat from the market? Some lipids, ghee is there, and different kinds of like peanut butter is there, butters. So all those are nothing but fat. Phosphorus and phosphorylated organic compounds in them. Some lipids. So some lipids have phosphorus and phosphorylated organic compound in them. These are phospholipids. If they have phosphorus group, they are found in cell membrane. Lecithin is one example. Some tissues, especially neural tissues, if you see, they have lipids with more complex structure. So neural, our brain and our nervous system, they have more complex structures. Now, living organisms have a number of carbon compounds in them which are heterocyclic rings. They can be found. Some of these are nitrogen bases: adenine, guanine, cytosine, uracil, thymine. All these are different types of nitrogen bases that are present, okay, in heterocyclic ring. Now, if you talk about when they are attached to sugar, they are called nucleoside. If a phosphate group is also found esterified to the sugar, they are called nucleotide. Adenosine, guanosine, I will explain you just a second. I will explain you this entire thing. Let me just read it now once. So adenosine, guanosine, thymidine, uridine, cytidine, all these are nucleoside. Okay, so you have something known as nucleoside and nucleotide. Nucleotides are nothing but, example is adenylic acid, thymidine acid, guanosilic acid, uridylic acid, then cytidylic acid. All these are nucleotide. Now what is the difference between nucleoside and nucleotide? And nucleic acids like DNA, RNA consist of nucleotide only. They have only nucleotide, that is known as our DNA and RNA. These are our, that is deoxyribonucleic acid, DNA, and RNA is our ribonucleic acid. They are present as genetic material. Now you can look here. See, when they have this chain, correct?
So nitrogen heterocyclic ring, see, you can see here, when they have two heterocyclic ring, two rings if they are present, this is known as, see, with nitrogen, the ring is not carbon entirely, okay, it is having nitrogen. So when it is having two rings fused together, that is known as adenine. When it is having only one ring, that is known as pyrimidine. When it is having two ring, that is known as purine. When it is having one ring, that is known as pyrimidine. Okay, now this purine and pyrimidine ring, basically this nitrogen bases, this is known as nitrogen base. When that is attached with this kind of chain again, with another chain containing oxygen here, this kind of ring, that is known as nucleoside, which is nothing but sugar. See here, you can see the structure of sugar, simple sugars. You can see here ribose, correct? That is why it is known as ribonucleic acid or deoxyribonucleic acid. If oxygen is removed from it, it becomes deoxy or ribonucleic acid. So this is a sugar ring. So if sugar is attached along with adenine or this, along with adenine or uracil, that is purine or pyrimidine. When purine plus sugar, ribose sugar basically, purine plus ribose sugar forms our nucleoside. Otherwise only this purine or pyrimidine, it can be purine or pyrimidine, okay, pyrimidine, these are known as nitrogen bases. So nitrogen bases plus ribose sugar gives nucleoside, and this nucleoside, when this nucleoside is attached with a phosphate group, when the nucleoside is attached with a phosphate group, it is known as a nucleotide. That forms our genetic material. Now let's talk about primary and secondary metabolites. What are primary and what are secondary metabolites? The most exciting aspect of chemistry is dealing with isolating thousands of compounds, small and big compounds, separating them from living organisms, determining their structure, and if possible synthesizing them also. That is most important thing about chemistry, right? Naturally whatever is present in our body, we are able to synthesize them, we are able to see the structure of them. That is one major part of chemistry. Now if we were to make a list of biomolecules, that list would have thousands of organic compounds in them, including amino acid, sugar, etc. For that reason, in section 9.10 we can call these biomolecules as metabolites. Metabolites means all the biomolecules, we can separate them as metabolites. In animal tissue, one notices the presence of all such categories of compounds shown in figure 9.1. So if whatever you see in figure 9.1, all these compounds are present. So you can identify, you can see sugars, you can see amino acids, you can see this fatty acids, you can see the, that is fats and oils, different types of fats and oils, cholesterol, phospholipid, and then you can also see the nucleosides, nucleotides, nitrogen bases, all in animal tissue. So these are called as primary metabolites because they are very important for the human body, right? They are mainly produced in our human body, they are mainly required for the function. But along with that, when you analyze a plant, fungal or microbial cell, you would see thousands of compounds other than these primary metabolites. Example: alkaloids, flavonoids, rubber, essential oil, antibiotics, colored pigment, scents, gums, spices, all these are known as secondary metabolites. They are not so important as primary metabolites. So while primary metabolites have functions and we know the roles of them in normal physiological process, we do not at moment understand the role of secondary metabolites. We don't know why they are exactly used in host organism. However, many of them are useful to human welfare. Example: rubber, drugs, spices, scents, and pigments. Some secondary metabolites have ecological importance as well. So in later chapter we'll be learning about them in detail. So if you talk about pigments, carotenoids, anthocyanins, all these are example of pigments. When you talk about alkaloids, morphine and codeine. When you talk about terpenoids, monoterpenoids, diterpenoids. Terpenoids are nothing but they have a terpene ring, okay? If they have one ring, it is known as monoterpene and diterpene. Essential oils are lemongrass oil, Toxins, these are poisonous things which are arbin and resin. Then concanavalin A, that is an example of lecithin. Then drugs are vinblastine, curcumin, all these are different types of medicines that are used. And polymeric substances are rubber, gum, cellulose, etc. Now let's talk about the biomacromolecules. Biomacromolecules are nothing but, I told you, they are found in the acid insoluble pool. That's why they are known as macromolecules. There is one feature common to this acid soluble pool. They have molecular weights ranging from 18 to 800 Dalton approximately. That is about biomicromolecules. If you talk about micromolecules, their molecular weights are ranging from 18 to 800 Dalton approximately. The acid insoluble fraction has only four type of organic compounds: proteins, nucleic acids, polysaccharides and lipids. These are the four types of things that are present in our acid insoluble pool. These classes of compounds, with the exception of lipids, have molecular weights in the range of 10,000 Daltons. So all these three basically, except lipids, if you see, all the three of them have molecular weight 10,000 Dalton or above. For this very reason, they are known as biomolecules, are known as macromolecules. Okay, so chemical compounds found in the living organisms are of two types. One is which have molecular weight less than 1000 Dalton. They are usually referred to as micromolecules or simply biomolecules, while those which are found in acid insoluble fraction, they are known as macromolecules or biomacromolecules. So biomolecules means micromolecules which are present in acid soluble fraction, whereas biomacromolecules means nothing but macromolecules. They are more than 10,000 Dalton, greater than 10,000 Dalton, whereas biomolecules are nothing but less than 10,000 Dalton. The molecules in insoluble fraction, with exception of lipids, are polymeric substances. Except lipids, all others are polymeric. Polymeric means they form polymers, they are big structures. Then why do lipids, whose molecular weights do not exceed 800 Dalton, come under acid insoluble fraction? Why they are present in acid insoluble fraction? They should be present in acid soluble because they are so small. They are less than 800 Dalton. Because lipids are indeed small molecular weight compounds and are present not only in as such but arranged into structures like cell membrane and other membranes. So when you grind a tissue, we are disrupting the cell structure. The cell membrane and other membranes are broken into pieces and form vesicles which are not water soluble. So they are not water soluble. Therefore, these membranous fragments do not form in the form of vesicles, get separated along with acid insoluble pool, and hence in the macromolecular fraction. Lipids are not strictly macromolecules. So lipids are not strictly macromolecules because lipids are present in macromolecular section or acid insoluble pool, but they are not macromolecules because they are only 800 Dalton, whereas I told greater than 10,000 Dalton what is present, that is considered as macromolecules. Now the acid soluble pool represents roughly the cytoplasmic composition. So whatever is present in acid soluble pool, that is roughly whatever is present in our cytoplasm. The macromolecular form of cytoplasm organelles become acid insoluble fraction. Together they represent the entire chemical composition of living tissues. So now average composition of cells, if you see, water is 70 to 90% in the cell because this is what is our cytoplasm. Cytoplasm is mainly composed of water. Proteins are 10 to 15%, carbohydrates are 3%, lipids are 2%, nucleic acids are 5 to 7%, and ions are 1%. So in summary, if you represent chemical composition of living tissue from abundance point of view, we can observe water is the most abundant chemical in living organisms. Now let's talk about protein. So proteins are polypeptides. Polypeptides means they are forming a peptide chain, linkage of amino acids linked by peptide bond. Okay, so lot of amino acids are linked together. Amino acids. So if you see